Hi, this is Professor Stugard, and in this video we are going to work on our second lab assignment. Alright, so I have two things open. I have RStudio open to a brand new project, and I also have our Blackboard. So our Blackboard, we are going to be creating visualizations based on that Palmer Penguins data set that we looked at last week. And we'll slowly build up more complicated visualizations, and hopefully we'll be able to actually glean some information from those visualizations that we create. So I downloaded that already. I have my RStudio project open. Again, the first thing we always want to do whenever we open up our new RStudio, we need to install Tidyverse. Now, remember that every time that we install Tidyverse with our new project, it is still external, and external means quotes. So install packages around quotes, Tidyverse, and again, that'll take a minute or two, so I'll pause the video. Actually, no, I will upload my Penguins 2 while I am doing this. All right. So let's see. All right. So again, it's still all installing. We have Penguins 2, which I can open up now as my markdown file, Penguins 2. And again, the instructions in this project, we will be exploring the Penguins data set included with the Palmer Penguins package and creating different visualizations. So what we're going to do for this one, inside each code chunk, we're going to, well, write the code that creates the visualization I asked for. So again, each code chunk is going to be distinguished in gray. We see that we have the kind of that same chunk here where we need to install and load the not just Tidyverse, but also the Palmer Penguins. Now, when you write in code chunks, if you want your code to not run, you comment it out, and you comment it by using the hashtag or number sign or pound sign, depending on how old you are, I guess. And so in the first in this first code chunk where I have packages, I have commented out those first two lines, so you do have to delete and delete. And notice how that changes color. When it is commented out, the whole line will be green. Again, that means that R is not going to run or compile that line. But now that it's back in black, well, yes, it is going to run that code for us. All right, so it looks like Tidyverse finished installing. So now we'll load library. And now that it is, again, in the environment, we don't have to put quotes. It will autocomplete. So we'll load Tidyverse. It takes about 2 seconds. There we go, all check marks. We'll install packages. Oops, it's external, so remember the quotes. Palmer Penguins. And again, that loads pretty quickly because there's not too much going on there. And then library Palmer Penguins. And again, it auto-completes for us. And now we can use it. Now, what I'm going to do first before we start doing these visualizations is I am going to knit this together as an HTML. Okay, we have to install and update some stuff because, well, that happens pretty frequently. All right, so I'm going to let that run. Now, the nice part about doing it like this is, so again, now I have an HTML. This is basically my instructions. And it might be easier to read this than the R Markdown file in RStudio, but again, that's up to you. But again, we're going to be installing the packages. We talked about the instructions already. One thing we want to keep in mind, and I wrote that here, again, that's the nice part about R Markdown is you can do italics and bold and everything else. We have to match the title of our column or our variable exactly when we create our visualizations. Now, visualization 1. This visualization should be a scatter plot that has bill depth along the horizontal axis and bill length along the vertical axis. All right, so now we're going to go back to RStudio, and I'm going to eventually, under visualizations, we have plotting number 1. Again, that my code chunk, we see that everything is greened out because I have my that in the hashtag, I guess, the insert code here. So again, we want to insert the code that's going to create that scatter plot into that code chunk. Then later when we compile and run and knit together our markdown file, if our code is correct, then that visualization will appear in our final product, which is very, really nice. So the thing is though, instead of having to knit that together every time, what we're going to do is play around in the console. And from that console, there we are going to, well, check out our visualization, make sure it works. Once we have what we want and it looks good, then we are going to copy and paste that code. So we're going to be using the Penguins data frame. And again, Penguins now exists. It is a data frame. We can look at it in the console because it comes with that package that we installed. If we want to explore it in our environment, we do have to store it there. So again, Penguins, store it in Penguins, and now I have a data frame there. Now I called it Penguins and Penguins. If you're like, oh wait, I wanted to make it Penguins data frame because it's in my environment and make it separate, we can always remove things from our environment with the function rm for remove. So if I do remove, and again, every function needs those parentheses, and inside the parentheses we put our argument. So remove from my environment, Penguins, and oh, look, now my environment is empty. And so I'm going to try that again. And here's the nice part. When you type things in the console, one of the nice parts about RStudio is that it remembers every command that you've typed into the console. So if I'm in my console, again, right, I clicked in there. If I just hit up, it brings back my last line of code. If I hit up twice, it brings back the second to last code. And again, you can go back all the way through your code and find things that you've typed and played around with in the console, so you can reuse your code and you don't have to necessarily do everything over and over and over again. We'll talk about that as we go through this lab. So I'm going to store it in my environment as Penguins DF for Penguins data frame. And again, it's just kind of a good habit to get into, labeling exactly what is what. And anytime we have a data frame, doing underscore DF for data frame is kind of nice. Make sure that when we look at it in my environment, oh, okay, I know that I'm dealing with a data frame and not like a vector or just a variable of some sort. But again, that's kind of personal preference. And again, we're going to be using underscores to create our objects. You can use periods, but we don't really do that much anymore. Although I know some JavaScript, they still like to use periods. But again, I prefer underscores, and especially opposed to camel case and a few other things. And just remember that, again, you always should have your object start with a lowercase letter, no numbers, no uppercase, no symbols, that sort of stuff. Okay, so let's actually get to the code that we want to do here, which is to create our scatter plot. Now, the first thing we need to do, well, my preferred way of doing things is to pipe in my data frame into my function. And you're going to see next week that that really kind of helps set up multiple lines of code that we kind of run together in kind of a way that's easy to look at. You don't necessarily have to do it that way though. But again, I like to start with Penguins, and then we're going to pipe. So the pipe is at % greater than %. Now again, what that's going to do, that's going to pipe my data frame into the function. Now, if I want to get to like the next line of code, which again is going to make it look nice, we hold down Shift and press Enter. Now, that gives me down to the next row, and it also indents it so that, again, it looks nice and will be formatted nicely for us. Now, the function that we use to create all of our visualizations is called ggplot. All right, and again, we can let it autocomplete for us. Now, ggplot. You can see that as soon as we type it in, and again, it kind of auto-completed for us, we see, okay, ggplot, the first thing is data equals, then mapping equals AES, and again, those are all of the arguments. Now, because we're piping the data in, I don't have to say what the data is. It would be redundant. If you don't pipe it in, you would have to start with the first data equals Penguins, and then you would do a comma for your next argument. But again, we don't have to do that since we're using the pipe feature. And And again, I think this just works a little bit better. So the next thing we need to do is the mapping. So mapping is, again, how we define our variables. So mapping equals, and we're going to use the aesthetic function, AES, which is, again, its own function, so its own set of parentheses. And you can see, again, as we autocomplete it, it's x then y. So again, we're basically creating what's our x-axis going to represent and what is our y-axis going to represent. Now, if you want, because we've already piped the data in, and we know that the first argument should be the mapping, you don't necessarily have to include that in your code. The program R is going to automatically know what it is that you're trying, that this is your mapping function, because it comes next. Data first, mapping next. And again, just the way these functions are structured in terms of which argument goes where. So my aesthetics. Now this I always do want to define with x equals and y equals. So x equals, and again, I prefer spaces because I think it looks cleaner. RStudio is going to ignore all of your spaces. So spaces and doing separate lines and doing indents, it's not necessary for the code to run, but it's very, very useful if anyone wants to look at your code later and try to figure out what it is that you're doing, which could also very frequently be you. So do future you a favor and always try to format your code in a way that's readable and understandable. Okay, so anyway, we're back to that aesthetic, which again is how I define what my graph is going to look like. And so x equals, all right, well, yeah, let's go back to the problem. So this visualization for visualization 1, a scatter plot that has build depth along the horizontal. So the horizontal is your x-axis, so I want the build depth. So build, and how nice it starts to actually autocomplete for me so that I make sure I type in my column perfectly correct. If you don't type it in perfectly correct, it's not going to work. And that is one of the frustrating things about coding and computers, is that it never does what you want it to do. It's only going to do exactly what you tell it to do. So we got to make sure we tell it to do the exact right thing. And again, you can type it in, or like I said, I suggest using the autocomplete features that come with RStudio because it does make life a little bit easier. So build depth is going to be along my horizontal, comma. And again, we separate every argument with a comma. And then my y variable along my vertical is going to be build length. So build length. All right, now I've defined what my graph is going to look like. And if I just run the code now, oh, well, there we go. We have a plot now over here in this bottom right-hand corner under plots. And we see that build depth is along my horizontal, build length is along my vertical, but there's nothing else there. And that's because when we use the ggplot function, we basically add layers one at a time. And that's really what we're going to be doing throughout this entire lab, is we're going to just keep adding layers upon layers until we get a really, really nice looking finished product. And I really think it's a good idea to, again, do these things one step at a time. Make sure something works and it's what you want before you try to do the next step. Do everything one step at a time if at all possible. All right. So, again, here's that nice part that I mentioned earlier. We just type that code in. We don't have my scatter plot yet. I can just hit the up key when I'm in my console, and it brings back that last line of code. So I don't have to retype everything over and over and over and over again. Now, again, once we're using this ggplot function, we are adding layers. That's the syntax we want to think about when we're making our visualizations. I want to add my layers. So we're going to put the plus sign, all right, add that layer, and then hold shift and press enter. That brings me down to my next line, and now I can add my next line. So now I want to add a scatter plot. So I know what my variables are on my graph. Now I want to add my scatter plot. To add my scatter plot, every type of graph is called a geom in R. And you'll see as soon as you start to type in geom, man, there are a lot of options. A, B line, area, bar, blank, contour, density, hexagon. It keeps going over and over. There's tons and tons of options. And that's really, 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 really nice because that means we can basically create any type of visualization we want using RStudio. We can make our visualizations look however we want. And we're going to be able to customize this in a million different ways as well. Now, for a scatter plot, so again, it's always going to be geom, then underscore, and for a scatter plot, it is geom point. And again, if you're ever confused, you can always, in your console, when it starts to autocomplete, hover over, and it will give you a description of that function. And so in this case, geom point, it literally says the geom point is used to create scatter plots. And the scatter plot is most useful for displaying the relationship between two continuous variables, which is what we have here with our build depth and our build length. And again, you can also see that it includes some of the arguments that we can define for this particular function. Again, really everything is customizable. Now, we're not going to actually do any of those right now. We just want to create a scatter plot. And if I press enter now and run that in my console, boom, there we go. Now we have a scatter plot with build length and build depth. And that's what my scatter plot looks like. But, ooh, look, something showed up in red. Something showed up in red. We removed 2 rows containing missing values. Again, anytime a warning message pops up, you should take the time to read it and make sure you understand it. In this case, it's saying that there's missing values. Missing values. If I take a look at my Penguins data frame that I have in my environment and I look, oh, I can actually see right here on observation number 4, there's no data for the length, the depth. There's actually no data at all for this penguin except for the year. So it's saying it ignored that line, and if we were to go through this whole thing, there should be another NA somewhere because it ignored 2 of them. I'm not going to scroll through 100 different, or I should say 344 different lines, but again, that's what's going on here. So we have our scatter plot. We have our basic scatter plot. This is what I wanted for visualization 1, so I hit up. Oh, view penguins DF. No, I actually want this one. So I'm going to copy and paste. So copy and paste, again, just highlight, Control C, insert the code here, so Control V, and that's it. And now again, it's in my, it is in my R Markdown file, which means it gets preserved. I'm going to save that particular line of code. And when I were to knit this together, what happens now, and I'll do this one more time real quick, is that once it actually knits together, my new my HTML file now includes my scatter plot that I just created. And again, right, that's how we preserve everything. I have it set up so that it preserves my code and it preserves any warnings we might get as well. Okay, so that looks good. We copy, we paste it, we saved it. That is visualization 1. Now we can move on to visualization 2. So we're going to, this visualization should be a scatter plot that is build depth along the horizontal. Still got that. Build length along the vertical. Got that. But plots each species in a different color. All right. So we're looking at 2 variables already. We have build depth and build length. We have 2 numerical continuous variables that we're looking at, and that's what we've plotted. Now I want to consider a 3rd variable, and that 3rd variable being the species. Now, when we're trying to do things with color, or again, trying to kind of customize our graph a little bit, when I do this, what I really want to think about is whether or not this is going to change the meaning of my particular graph. And in this case, it will. Instead of just having a bunch of random black points, we're going to have different colored points representing the different kinds of species. So going back to, again, that line of code, and again, I'm actually in the code, the line of code that I can type things into here in my console by hitting, up. Because this is going to actually affect the way that I interpret this graph, I want to put it inside my AES function. I want to put it inside my aesthetic function, which means I need to add another argument. So comma, space, and again, what I want to do is change the color. So color equals, and again, I'm doing it by species. So species. And if I run this code now, I think I'm getting a better understanding of what's going on with this particular data set. Now when I separate it by color, I can see these patterns much easier. There's definitely a pattern where the size of the bill is different based upon the different species of the penguin. I just was able to learn a lot more about my data set by taking a look at one more variable. So now I'm actually considering 3 variables in my one scatter plot, my one graph. The bill depth, the bill length, and then the species and how they all relate to one another. All right, but that's good. That's exactly what I wanted. So I'm going to hit up again. My code worked. That's what I want to see. So I'm going to copy and paste this and insert that here for plotting number 2. All right, now we'll move on to visualization 3. I'm not gonna reknit this together. Now, just to kind of mention, if obviously I'm going to quickly pause the video, go back, rewatch it, take your time. Again, this is something that we would expect to do over the course of an entire hour or so in a computer lab. So again, take your time and pause the video as necessary. So, step 3. Now that I actually do have a nice visualization that tells me a lot about this data, well, I need to have labels for it. I want it to look nicer. And again, we can see here that bill underscore depth underscore mm, that's not really that nice looking. I would be embarrassed to hand that in as a professional looking report if I was a data scientist. I need my graphs to look nice and clean, and they should look very professional, and who's ever looking at my report should know exactly what it is I'm trying to get across. So, again, we have our code. I can edit it here in the console. To add labels, we add another layer to my graph. So to add another layer, plus, and then Shift Enter, brings us down to the next line. And then to put in labels, it is the labs function. And again, as soon as we do labs, which comes with ggplot2, good labels are critical for making your plots accessible to a wide audience. So ensure that the axis and the legend display the full variable names. And again, we can see we type in, we can add titles and subtitles and captions and tags and a whole bunch of other fun stuff here. All right, so it's a function. We need our parentheses. And again, we're going to basically fill in, we want to have all of these for our different labels on the graph to make it look nicer. So I'm just going to copy and paste each one at a time. So the title, bill depth and length, so copy. And inside my labs function, so inside that parentheses, I'm going to paste it. So that's my first one, and then a comma. Again, remember, we're going to separate everything with a comma here. Now, I'm going to be doing a lot of different labels, and again, this can kind of get, well, kind of ugly and not look so nice. It's going to run even if your code looks ugly, but to make it look a little bit nicer, I'm going to do Shift Enter to get down to the next row. And again, next row, tabbed over because I'm still inside of my function. And now I will copy and paste my subtitle. Copy and paste, so again, where I was. Then a comma. Again, one of the most common mistakes is not including a comma to separate your arguments. And again, by doing this on new lines, I can see every one of my lines ends with a comma. And again, if you run into an error, which you're going to run into errors, that's a really good thing to check for first. So I have my title, my subtitle. Now I want to label my axes. So my x-axis was bill depth. But again, instead of doing the underscores and stuff, I'm going to try to make it look a little bit nicer. So bill depth for my x-axis. My y-axis is the bill length. And again, I'm including my units, but I'm including those in parentheses instead of, this kind of underscore. And then for my legend, I know that I was doing the color by species, so again, copy and paste, and oops, I'm already seeing that I made a mistake. I forgot to put a comma at the end of my y equals bill length line. And again, by writing clean, neat code, you'll find your mistakes much quicker. Okay, so that color for species, and then one more line because we have one last thing for a caption. because we want to include where our data came from. So source. All right, let's see if this works. So I run my code, and uh-oh, unexpected input. Okay, so what happened here, and again, this is a very common mistake that happens when you copy and paste code. Microsoft, so I'm going to hit up so I can take another look. This last one, you can actually see, if you just copied and pasted, that the color for my quotes is different. And it's the quotes that kind of curl in, which is how Microsoft Word formats it, and a few other things format it. Unfortunately, that is technically a different symbol than the normal quotes, and Microsoft Word just changes it to make it look nice in the background without you even noticing. So copying and pasting code can lead to lots of issues. So what I need to do is put in and retype those parentheses. I'm sorry, not the parentheses, retype those quotation marks myself and redo that. And so again, that is when I read my error in red, unexpected input in, and again, we see here that captions was where that issue showed up. All right, so let's see if this works now. Hopefully it does. Nice. So again, I still get a warning message, but it's that same warning message that it removed those 2 rows with the missing values back when we did the geom plot, or sure, the geom point part of this graph. And again, hopefully now we can see, well, yeah, this looks much nicer. This is a much better visualization because everything is very well labeled. Someone's going to look at that and they should have no questions at all about what's going on. So I'm going to hit up again, bring back that last line of code, and I'm going to copy and paste that entire thing into plotting 3. All right. And again, the nice part is our markdown, anything that's a quote or going to be a label of some sort gets put into green because it's inside my quotes. And there we go. Now my code is preserved. Okay. On to visualization number 4. So for visualization number 4, this visualization is a scatter plot with build depth along the horizontal, build length along the vertical, but plots each species in a different color. Done. This visualization also has to have the following labels. Now, we did all of that. The last thing I want to do is change the colors used to add accessibility for the colorblind. So by default, RStudio picks red, blue, and green as the colors that they use for the scatter plot. And you can define each color individually. You could pick each color if you wanted to, one at a time. If you really, really, really want to customize it. But again, unfortunately, red, blue, and green, that is literally the colors that are indistinguishable when you're colorblind. So someone who's colorblind is not going to get much use out of this particular graph. Not only that, if I were to print this out in black and white, it kind of all looks the same. So one of the really, really nice things is that somebody else wrote another function to make this accessible for the colorblind. And we just add it as another layer inside of our ggplot. So at the end of the code that we just typed with all of our labels, I add a plus sign, hit Shift and Enter, and now again, notice where the cursor is. It brought me back over one indent. So again, it keeps all my labels nice and organized. Everything that's part of my labeling is part of that one chunk. And now I'm back over where my next layer is going to be. And I'm just going to copy and paste this without the tick marks. Do not copy and paste the tick marks, so just the plus. Oh, I already put the plus in. So, oh, I'm sorry, just copy and paste the scale underscore color underscore Veridis. Copy, paste, and Veridis D. And I can run my code. And now it changed it to colorblind friendly colors. We have yellow, teal, and purple. And someone who is colorblind will be able to distinguish those colors. So that is a much more accessible visualization to use. So we'll copy and paste that. So copy and paste that into number 4. And And there we go. All right. Now, this is not the only visualization theme that there is. You can actually go through and play around with a little bit. If you do scale, underscore, you'll see that they actually have a bunch of different ways in ggplot to basically choose your colors and your gradients and change a whole bunch of different things. And in fact, scale, color, underscore, again, we can just literally change the colors and the scores. And then there's, again, like I said, we did Viridis D. There's also B and C. So B, bind scales. Why did that decide not to work? Oops. I know why. Because those are for different types of data. B and D for different types of data. So again, you're going to get some error messages, but again, there's different themes and different ways we can customize this. Okay. Moving on to visualization number 5. So, everything that we've had before, build depth along the horizontal, build length along the vertical, species in a different color, we have our labels, we added the accessibility. The last thing I want to do with this particular set of visualizations is I want to adjust the size of my points so that each point on my scatter plot is a little bit bigger. But because there's also overlap, I want them to be kind of transparent so that I can actually see when they overlap. And when they overlap, if you want to change the transparency, that is called alpha. So I'm going to bring up my last line of code. So I'm going to change those two things. Now, again, yeah, you could make an argument that changing the size of the dots and changing the transparency might change the way you understand this graph, and that is valid. It's not really changing the data or how everything is set up. So these two options are going to be changed inside of the geom_point function. So if you go up here to the geom_point part of my whole line of code, this is where I'm going to adjust the size and the transparency. So size equals 2. Again, that's just an argument. Very straightforward. Size equals 2, then a comma, then a space just to make it look nice. And then I'm changing my transparency, which is alpha. So alpha, spell it right, alpha equals, and then again, alpha goes anywhere between 0 and 1. It's like a it's like a proportion. How much do you want it colored in? And we're going to do 0.5, which is half. So it's at like half transparency. And so you can see now they're a little bit bigger. They're a little bit more transparent. So we can actually see where there's overlap. And we can definitely see some pretty big overlap with some of these penguins. So obviously our data is actually much more clustered towards some centers for each species of penguin than we probably would have been able to see otherwise. All right, so I'm going to copy and paste that code in here for visualization 5, and plotting number 5. And again, if you want to go around and play with the different alpha options, so 0.1, what does that look like? Well, that's so faint you can't really see it. At 9, well, that's so filled in that, again, you can't really see the overlap. And that's why I picked 0.5. 0.5 is kind of a nice one in the middle for this particular data set. But again, like I said, play around with this. Don't just necessarily do exactly and only exactly the particular things that I'm looking for. Take some time and actually like play around with it. Use different ideas here. All right. So now we're going to shift gears a little bit and we're going to do faceting. So faceting is really, really helpful as well when we want to look at multiple variables at a time. So like I said, what we've been looking at here, we have 3 variables, the build length, the build depth, and then the species. We can look at 3 variables at once, which is really, really nice. I can actually expand this and look at more variables if I wanted to as well, with lots of different choices. Facet is going to create a bunch of different plots where the plots are now separated based upon some other variable as well. So Facet Grid creates multiple visualizations using 2 more variables, and Facet Wrap adds 1 more variable. And again, we'll kind of talk about this as we go through. So, what I want for visualization 6, a grid of scatter plots that have bill depth along the horizontal, so we still got that, bill length along the vertical, still got that, and they should compare the variables species by row and sex by column by including this following code. Now, I have in there that you do not need to include the labels, or we do not need to filter by color for this one. So I'm going to get rid of all of that for now. We can certainly keep that if we wanted to. And we don't necessarily need to mess around with the size or the transparency here either. We're just kind of going back a few steps, right? Where, again, this is our visualization, and actually, yeah, we don't need to filter do the color by species either. So I'll get rid of that. So we're kind of back to that first visualization that we started with. Now, I have the code here, so again, we can just copy and paste it without the tick marks. So I'll bring up my last line of code, add the plus sign, shift enter, add that line of code, facet grid, species sex. Let me run that code now, and here we go. All right, so again, we can kind of see the clusters now, and we can see, all right, well, things are definitely different based upon the species and the sex. So there's definitely a difference between male and female penguins for each of these different species as well. And we can definitely see that those clusters get much tighter together, which means that, again, the gender and the species of the penguin seems to indicate, the size of the different bills. So, again, this is the code that we're going to preserve. I want to go back when we did that facet grid and we just copied and pasted. So this part that's inside the parentheses, that argument for species and sex, this is kind of the way that we do functions in R. So instead of like the y equals x, or the f of x, or y equals f of x, or whatever, instead of an equal sign, we use the tilde. So again, the tilde would be the shift tick mark right next to 1 on the top part of your keyboard. And the way it works, and we can see this here, so species, which I put first, goes along your vertical. And then sex goes along your horizontal. That is the way this is set up for all of our different types of functions. And we're going to talk about this. It'll come up a few other times throughout this semester. When we're working in R, it's always the response variable, your independent first, then the tilde, then your explanatory. All right, so it's response, tilde, explanatory. So again, what we're doing here, it is response or vertical, tilde, explanatory or horizontal. All right, so again, that worked. So hit up, and I will copy and paste. One thing I probably could have mentioned a little bit earlier is that, again, I always hit up to copy and paste after I've run my code so that I can copy and paste that. The reason for that is that if you look at the difference here, see how they put plus signs on each of these lines? Basically, the plus signs let us know that we are still using the same line of code and it ran it this way. But if we copy and paste it with the plus signs, your code is not going to run in R Markdown. You cannot include those plus signs in your code. So if you go back through and double check all of your work, make sure that inside your code chunks, inside that light gray section, you don't have any out of place plus signs. Because again, it would be plus sign, which that's not really going to work. RStudio is going to get very confused with that and give you error messages. All right, so we're almost at the end here. The last visualization is going to be a facet wrap for species. So like I said, we don't need to include any of that other stuff. So we're basically going to take this, and instead of facet grid, which lets us look at 2 variables, I'm going to replace that with the facet wrap. And again, we have our tilde here, which means that, again, that response, tilde, explanatory. So now the explanatory is just the species. which means when I do my facet wrap, instead of making a grid, it is just going to do them all in a row. Now, if there was a lot more options, and again, we only have 3 options for species, but let's say there was like 10 options, it would wrap it and keep going down the down the whole plot screen. So again, we can see that we are definitely grouped. We have clusters here for each one of the particular species. So I'm going to copy and paste that code. And that is it for this particular lab. Now again, on your own, you should start playing around with this and be like, well, wait, can I change the color? Can I add labels? And yes, you absolutely can. And play around with that a little bit. Try to make this look even nicer. But for this particular lab, you're pretty much done. The only thing you need to do now is change the author and the date. And I guess you could call it penguins too if you want. But absolutely change the author, change the date to today's date, and then knit it all together. So I'm going to knit this to PDF, because again, that's how I want it submitted, is the PDF format. We'll give it a second to load and everything. And again, if you get an error message, which is very common, it should tell you where that error is. When you have errors, it will show up. There'll be little X's in your code, in your markdown file, telling you where that error is, and you will get error messages. It's part of learning, is getting error messages. And hopefully you can figure it out. If not, again, don't hesitate to email me. The nice part is, again, let's label this Penguins 2 visualizations, or something along those lines. And again, if you're having trouble and you keep getting error messages, the nice part about RStudio is that if you go over here, where you're, oops, hold on. If you go over here, at the very top, we have RAM, we have the settings thing, and Professor Stugart. If you go to settings and you click access, who can view this project? Well, by default, it's just you. If you click everyone and you change that setting, what you can do is actually send me the link. Just literally copy and paste that link, send it to me in an email, and I get to open a copy of your project, and I can help you find out where your error might be, and then I can hopefully share with you how to fix your code so that it runs. All right, so we had our visualization. Here's my PDF. And again, all my code, my visualizations, here they are. And again, you can absolutely customize all of this. You can change the size of these inside your R Markdown, and there's a bunch of other things we can do. We're not going to worry about that right now. Again, I just want to really focus on one thing at a time, and I'm sure that this is plenty for week 2 of this course. But again, double check, make sure everything looks right. Make sure everything came out the right way. Okay, good. This is visualization 3. We have all of our labels. Visualization 4. Now it just is in the correct colors. Visualization 5. A little bit bigger, but a little bit more transparent. And we have our facets and everything else. Okay, that looks really, really good. So once you're happy with how all of that looks, make sure that you download it and then save it to somewhere that you understand on your computer. Once you have it saved, then again, on Blackboard, use the submission link to submit it to me. All right. Hopefully that went well. If you have any questions, again, like I said, please don't hesitate to reach out. And as always, take care of yourselves.